Today I'll try and cover these major points, and you'll notice that what's missing from there is reverse transcription. The standard of care is Antiviral, tenofovir, and we heard from Dr. Mitsuha on the first day an update on nucleotide analogues and hepatitis B, so I won't cover reverse transcription in the interest of trying to sort of keep on time and in context. So I think, being a virologist, I like to sort of start with a picture of the virus. I think this is hepatitis B virus in serum, and it's interesting to always go back to the old photos that, where you see that hepatitis B virus is really a diverse beast. You'll notice that the electron microscope has revealed both full and empty particles, and also donut-style particles, as well as sort of very weird sort of, I guess, pineapple-y type, being in Hawaii, pineapple-type particles. So the point being is that we've got structures here that don't resemble a clonal population of virions, and it's sort of, these old photos really need to go, we need to go back and reacquaint ourselves, because the point being is that as we've now learned with the major receptor and the minor receptor, that how hepatitis B virus gets in and gets out, that there's multiple cycles of not just entry but re-entry. And the virus is basically, from that previous electron micrograph, I was trying to capture the concept that as well as having sort of a clonal population or a dominant quasi-species pool, there are a lot of variants that are very atypical for a virus such as hepatitis B. And these include not just splice variants, which we discussed very briefly last night, but also full-length RNA. And why pregenomic RNA is packaged and secreted is not really known yet. So what this sort of picture, this cartoon is trying to capture is the traditional or the classical life cycle of HBV, of entering through its major receptor and the genomic DNA being converted to the minichromosome. Transcriptional products are made from host cell machinery, and reverse transcription occurs here, and then you get the generation of lots of HBVs. This is obviously interrupted by specific nucleotide analogues at this step, the reverse transcription, and we see a dramatic drop in HBV DNA. The infectious potential of HBV has been recently recognised with the realisation that RNA is packaged and secreted, and so that you get these multiple sort of cycles of reinfection. And I think an unmet or an unknown question at the moment is the role of these products that have been packaged and secreted into the extracellular environment, if they've got the right sort of viral proteins to sort of be allowed to sort of come back in and reinfect the hepatocyte. So the message here is that the virus is sort of in a cycle of, rather than cell-to-cell spread through intercellular junctions, it's extracellular sort of secretion and then re-entry. I think further consolidating this concept of the virus coming in with a diverse range of sort of materials was a very nice study done by the Gilead group from Virion and his colleagues at Gilead, which tried to sort of identify through deep sequencing and RNA-seq the sorts of timing, if you like, of the particular RNA transcripts of the virus. And what you can see here is that before infection, there's obviously no specific markers of RNA, but within 4 hours of infection of HBV, this is cell culture work, but the message is clear to me, the major transcript identified was the X transcript. And it wasn't really until sort of 7 to 10 to 13 days post-infection that the rest of the actual transcriptome of HBV was actually detected. So it looks like these particles that we were seeing in the extracellular space, not only do they have the genomic DNA and pregenomic full-length RNA, but they also have the X transcript as well. So let's have a look at the entry process into hepatocytes. There are two major receptors now. There's the heparan sulfate proteoglycan receptor. This is considered a low affinity receptor, and the A determinant of the surface antigen is the binding domain of that ligand, and the major high affinity receptor, the NTCP protein, discovered by Wenwei Li in Beijing, interacts with the N-terminal part of the pre-S1 region, the NTCP binding domain. So what we can see here is that the sites for inhibition can be heparin itself and sulfation inhibitors, glypican-5, siRNA, which is the major proteoglycan that's present here, and the also antibodies to the A determinant. These are the vaccine antibodies, and it's quite interesting to note that the vaccine antibodies are associated with partial neutralization and protection. And here on this side, we've got the NTCP agonists such as bile salts, cyclosporin, ritonavir, and the major and Macludex itself. And Macludex is acting as a competitive inhibitor of the binding site between NTCP and the N-terminal of pre-S1. And so antibodies to the N-terminal part of pre-S1 are known to block interaction between the pre-S1 and the NTCP receptor. So the second part of the major targets towards control of hepatitis B is the cytosolic transport of the nucleocapsids. We know that in primary infection, mature nucleocapsids are transported via the important alpha and beta proteins, and the nuclear protein 153 delivers these nucleocapsids to the nuclear basket where they uncoat and they enter the nucleus through the nuclear pore. Genomic rcDNA enters the nucleoplasm, and at this stage the polymerase is covalently bound to the minus DNA and then is converted to protein-free rcDNA. HBV RNA species, presumably as we showed in that earlier cartoon from the Gilead group, this is where xmRNA might be released and translated in the cytoplasm to give it an immediate early protein interaction. Now, this is an opportunity for where core active compounds could block the nucleocapsid uncoating and release of the rcDNA. Now, during persistent infection, we've initially was discovered in the duck model, but the intracellular conversion pathway was originally described as a major source of replenishment of the minichromosome, but probably in human HBV, the view is moving towards it's really a low-level recycling of nucleocapsids. As I said, that the major pathway is through multiple rounds of reinfection, and this is what really drives and replenishes the key intermediate. So really, at this stage as well, a nucleocapsid disassembly with a core-busting compound would actually be very effective. Really, the sort of guts of the virus, the sort of engine, if you like, is the what I call key step 1 and key step 2. Key step 1 is conversion of the genomic relaxed circular DNA into the CCC or minichromosomal DNA. And this step here really involves at least 6 steps, presumably all host cell driven, because there's been no activity associated with the viral's enzymatic machinery that seems to be able to do all of this. And a number of TDP2, tyrosine diphosphorylase enzyme, host repair enzymes that will probably typically be involved in the conversion of this really weird damaged-like DNA structure into the covalently closed circular molecule needed to form a minichromosome. And so these steps really probably are all linked into the DNA damage reaction machinery part of the host cell. And once this genomic DNA is repaired and functionalized, converted into a minichromosome, and then using host cell machinery, pre-genomic RNA is generated, shipped out to the cytosol, and then replication complexes are assembled, and through reverse transcription the cycle is completed. So this is sort of like, if you like, the generation and the processing of the viral minichromosome. So cccDNA is what the sort of molecular biologists call this particular structure that's dominant in the nucleus, and really, it's non-physiological itself, but is now known to exist as a viral minichromosome, first shown by Thomas Bock many years ago with electron microscopy. And we confirmed that in various animal models in our own lab, and identified particular replicative intermediates, or if you like, topological states of the cccDNA minichromosome. If you have an SV40 background, you know this is a 2-dimensional chloroquine gel and can resolve all of the DNA species typically seen in the nuclear cut into open circle, double-stranded linear, and here the topoisomers of covalently closed circular DNA, depending on their torsional state, can be resolved into two populations of fully chromatinized or half chromatinized cccDNA molecules, shown for you diagrammatically here as the closed structures with fully populated nucleosomes, or the open, and this represents this little group down here, or the half complement of nucleosomes B1, B2, which is shown for you here in the nucleosomes 10. So when you work out that this is 3,200 base pairs, there's 20 of these, you realize that the nucleosomal DNA is 150, then you can straight away realize there's a structural difference between host cell chromatin and hepatoviral chromatin in the sense that there's no linker DNA. There's no H1 histones. There's just no room for them in the structure. So there's at least, I think, the first sign that there is a structural difference between the viral minichromosome and host cell chromatin. Now this data was built upon what we now call the Levero structure. Sorry, Massimo, but that's where he proposed and has shown over the last few years that both the core antigen and the X protein are key components of the viral minichromosome, obviously along with transcriptional factors that are important in the driving of the transcriptional machinery of the HBV. Now shown for you here is the high replication open phenotype of or open structure of the minichromosome, where X and positive transcriptional factors and acetylation factors are associated with the structure, giving it an open transcriptionally flowing format, whereas in the core and X are critical components of this structure. In the closed structure, where you've got the deacetylation enzymes and the methylation enzymes, and the equivalent of the HBx mutant HBV, it's a low replication phenotype, and as I said, closed and sort of hyperchromatinized. So we now know that HBx blocks the methylation complex. I'll show the data for that in a second. But core antigen has a relatively amazing sort of DNA species-specific binding area and binds upstream from the basal core promoter into a CpG island 2 region, and really regulates and is a structural component of the minichromosome. But the critical message is that X knockout viruses result in transcriptional arrest or transcriptional inactivity of HBV. And the reason for that is that X is an important recruiter of enzymes, lysine demethylase 1 in particular, which demethylates the mark on histone 3, lysine 9, reduces the methylation status of this particular negative regulatory histone mark. And so in the presence of X, it becomes a transcriptionally active viral promoter, and it attracts acetyl transferases and removes deacetylase activity. And in the absence of X, obviously methylation markers come in and negatively regulate the minichromosome. Gilead put this together with Michael Strubens from Geneva into a very elegant understanding of transcriptional control and regulation by the X protein and the host cell restriction factor structural maintenance chromosome 5 and 6, which really belongs within a very large structure called nuclear domain number 10. So the model that we've currently developed now is the thinking about the virus comes in, it's got X, it generates an active X protein, and the whole point of X is to inactivate this structural maintenance chromosome 5 and 6 complex through ubiquitination through E3 ligase. And it does this in a very obviously rapid way, and hence why that first slide I showed, or that earlier slide I showed, was so important, that the virus is able to control host cell transcription immediately on entry and penetration into the nucleus. And so in the presence of X, you get inactive ND10, SMC56, but in the absence of X, you get an active cccDNA. And so this balance, if you like, this critical interplay between the X protein and these host cell restriction factors like SMC56 really determine the outcome of the transcriptional state of HBV. So in terms of actually targets, HBx inhibitors, in order to silence the minichromosome, is a very reasonable chemotherapeutic approach. the crystal structure or part of the crystal structure of X is known through and also the DDB1 ubiquitin ligase complex. So this interaction between HBx and DDB1 can be studied, and obviously people are looking at small molecule inhibitor screening. Obviously X as a target is a very important one. As I said, as part of the Rivero structure, the core protein is also a key component of the viral minichromosome. And it seems to bind with some specificity to this upstream CpG island number 2. And these are electron micrographs again from Thomas Bock, and showing the interaction of the core protein probably more as a structural rather than as a functional sort of component of the HBV minichromosome. I think that the realization and the identification of the core protein in the minichromosome really laid the basis for the important observation and work from Ulla Protzer about the role of the APOBEC apparatus in showing for the first time a selective specific degradation of a viral minichromosome. And Ulla's group was able to show that through the lymphotoxin beta receptor protein interaction with its receptor, that APOBEC3B is activated, and APOBEC3B is able to recognize something that's uniquely viral, and that's the hepatitis B core protein intimately associated with the viral minichromosome. And by doing that, you get deamination of the viral minichromosome, uracil excision, and degradation. So this was really a very exciting piece of work because it showed for the first time selective destruction of the viral minichromosome by exploiting an elegant cellular pathway. Massimo Oliveira's group had also shown that interferon alpha can do a similar thing through APOBEC3A, but hadn't quite put together the link with the cccDNA through the core protein. So I think what we put together, it really tells a very elegant story about selective destruction. So if you're thinking about if the core protein is an important part of the viral minichromosome, then obviously targeting nucleocapsids would be, you would predict, could actually be an important strategy. And we'll come back to this in a few minutes, but just to sort of put it in context of the presentation, there are a number of capsid inhibitors being developed by companies at the moment. The sulfonamide, sulfamoyl benzamide derivatives, obviously taking the lead of their better bioavailability, and the HAP compounds, which is a long history going back from Bayer back into the early 2000s. Interestingly, in the clinical studies that have been reported to date, the most important effect is, again, having HBV on HBV RNA. So the capsids, obviously, as well as blocking the capsid assembly, it also and reduces DNA. Circulating HBV DNA certainly reduces HBV RNA. So to sort of start to bring some of this together, this was presented a bit by Ulla last night at during her plenary. How are the various groups thinking about how can cccDNA be cleared? This has sort of been called the holy grail of hepatitis B therapeutics. And so there's basically two camps. There's the immunological camp and there's the virological camp. So out of respect to my immunological colleagues, I put you first. And so there's the cytolytic mechanism, which is presumably mediated through a destructive pathway, presumably from CD8 T cells, where you see replacement by infected hepatocytes and dilution of cccDNA content, as shown for you here. Or you can get replacement of lyased hepatocytes from uninfected precursor uninfected hepatocytes. Professor Cesari's group showed elegantly some years ago that the combination of the antiviral cytokines, interferon gamma and TNF-alpha, can actually cure non-cytolytically cccDNA containing hepatocytes. The virologist has a bit more pragmatic approach, and the view is that we like to sort of think about purging and killing. So direct purging of all the pre-existing cccDNA was shown by Summers and Mason some years ago, showing how you can selectively destroy topoisomers. The other approach taken by Newbold and our group was to permanently silence the cccDNA and drive the minichromosome into the condensed hyperchromatinized form and transcriptionally inactive. And of course, the third approach would be by specific or selective cccDNA targeting using small molecule antivirals, such as these disubstituted sulfonamides. And in the era of these molecular-based therapies, we can now sort of think about therapeutic approaches where we can cleave cccDNA molecules via sequence-specific endonucleases, and a number of these have been published recently, and we'll hear a bit more about these during the program as it progresses. The zinc finger nucleases, the talon structures, but really there's been a lot of interest and investment into the CRISPR-Cas9 technology. And Christoph Seeger published an elegant paper some years ago showing that indeed using this platform that you can actually knock out genes, can inhibit HBV replication by 8-fold, and showed specificity to the cccDNA form from the recruited Cas9. And this inactivity was basically achieved through non-homologous end joining recombination. Interestingly, the inhibition activity of the CRISPR-Cas9 platform that Christoph developed was unaffected by interferon alpha when used in combination, and a number of groups have now independently confirmed the seminal observation. For the presence of time, I'll remove those. So I'll jump over reverse transcription and now look at viral transcription generally from the minichromosome. So we've covered sort of the genomic replication, its disruption by nucleoside-nucleotide analogues, and now we'll have a look at the packaging of the pregenomic RNA. So I snuck this one in because this has been a recent sort of target in terms of the context of a direct-acting antiviral. Now, I'm not saying that Rig-I is, but we do know that the Rig-I does actually have an anti-HBV activity, but studies from Charlie Rice's group showed that it also, as well as driving a type 3 interferon response in hepatocytes, Rig-I can actually, by recognizing the 5' epsilon structure of the pregenomic RNA, interfere with actually the reverse transcription reaction. So you might remember that the reverse transcriptase is generated from in cis from its own pregenomic RNA molecule, folds back onto the epsilon structure and primes reverse transcription on the bulge, and is then subsequently translocated. Rig-I, by recognizing this pathogen-associated molecular pattern, or this PAMP of the 5' epsilon RNA, actually blocks this whole reaction. And this has actually been shown recently by the Springbank group they invented this molecule called SB9200, which has been given the name Inarigivir. It's a small dinucleotide of uracil and adenine held together by a phosphothiazide bond. And this seems to enhance the interaction of Rig-I to the 5' epsilon structure on pregenomic RNA. I'll present the mechanistic stuff this afternoon on behalf of Springbank, so I won't discuss this any further, but this is an exciting new approach in terms of a dual mechanism of action, sort of not only a DAA but also a interferon inducer. In the clinical studies, the drug had an amazing effect on, this is a 25 milligram from the clinical trial Achieve study just been presented at ASLD last few months ago, a 3-log reduction in the HBV packaged RNA and a 2-log reduction in the core-related antigen, mainly in e-antigen negative patients. And there was a modest, a significant decline, but only one, not a matching 1-for-1 log reduction in HBV DNA. It was only 1 log reduction compared to the 3-log reduction in RNA. And the fact that the e-antigen negative patients do better than the e-antigen positive patients is not clear at this stage. Oops, sorry. So the other interest in the terms of transcription events in terms of HBV life cycle can be found in the RNAi studies. Initially, successfully sort of proof of concept given from the Arrowhead group. Most people will be familiar with the RNAi mechanism in terms of the double-stranded RNA molecules are introduced into the cell and they're processed through dicer into 19 to 21 double-stranded RNA molecules, are then separated on the RISC apparatus with complementary pairing from the target strand, the trigger, and then allowing cleavage of the target RNA to promote therapeutic silencing. So this whole RNAi process has been around for a while, but the problem was always in terms of delivery. Arrowhead initially started with a DPC, a dynamic poly conjugate, and unfortunately hit some toxicity issues last year. But before that happened, they were able to show some important proof of concept studies that in the single dose cohort 7, they were able to get a 1 to 2 log reduction in surface antigen, which is the only convenient readout in the extracellular space. And in the extension cohort, they were able to achieve a 1 to 3 log reduction shown for you here very nicely in terms of some patients. As I said, unfortunately, the study was stopped because of toxicity with the delivery apparatus, and Arrowhead are coming back with some new delivery platforms. We've discussed this extensively through this meeting, the fact that these studies, as well as providing proof of concept for RNAi technology and that it could actually be a profound antiviral for the hepatitis B proteins through the RNAi process, showed that in E antigen positive, these are the chips that were used to sort of develop their therapeutic strategy. In the E antigen positive animal here, most of the RNA comes off a typical episomal transcript, and the polyadenylation signal behaves itself, and 90% of the transcripts are indeed pure or true to the poly-A signal. This is one here where there's a fusion sequence with the host. But in the E antigen negative animal, studied here by the PacBio's single molecule real-time PTR sequencing, that there was 90% of the RNAs were actually fusion transcripts with host cell sequence. So that we can see that this has been an important contribution to our understanding of the pathogenesis and natural history of chronic hepatitis B. Now, I'm missing a slide. Oh, sorry, yes. The sort of take-home message from these important studies was that there was certainly most of the DNA in the E antigen positive animals was cccDNA, 500-fold less cccDNA in the E antigen negative animals. And the DNA profile in the E antigen negative chips was consistent with a high proportion of integrated DNA. So I think in terms of therapeutic approaches, the direct antiviral effect on serum surface antigen and E antigen in those positive patients, as well as a core-related antigen, was quite significant, and there was no rebound to baseline from NADA, which was quite interesting. So the successful attempts by Arrowhead to sort of show proof of concept have been confirmed by Arbutus, and this data was presented last month at the AASLD in their phase 2A study, looking at their ARB 1467. Their dosing was much lower than what was achieved by Arrowhead, and using, you can see here, and they were able to show both E antigen positive and E antigen negative patients showed after 3 injections, 3 infusions, to achieve a significant reduction in surface antigen of up to a log. So I think that the scene is set now for the RNAi chemotherapeutic approaches to really sort of drive forward a very successful antiviral approach in hepatitis B. Now the second last target I'll look at is the HBV core protein. The core protein is really like a gatekeeper of HBV replication. It regulates not only assembly and amplification of cccDNA, but also the maintenance of the minichromosome. It even has some immunological effects as well. So in terms of its structure, it's got, it forms dimers, and these dimers assemble into the nucleocapsid. And people recognized many years ago that the core dimer interface was a potential target, and both from the HAP molecules as well as from the phenylpropanamide derivative series, that this core assembly reaction around the pol pre-genomic RNA complex could actually be a very useful target. And so I'm sure we'll hear about over the next few days a number of compounds that are in evaluation of these core active molecules. I'll present a little bit of clinical data because I think it's important to sort of know what we're to expect. And this is a GLS4-JHS, which is a HAP molecule from China. It was again presented at AASLD last month. And what it shows is that they ritonavir boosted their molecule, 100 milligrams of ritonavir versus 120 and 240 of the HAP. And they compared that to entecavir in the red line, and here's the DNA response. They're achieving a reasonable 1 to 2 log reduction in HBV DNA with the ritonavir boosted GLS. But really, I think that I'll draw your attention to the E antigen effects and the surface antigen effects. So the surface antigen effects are zero, unfortunately, so they've been unable to show any impact on the circulating surface antigen and likewise circulating E antigen. So even though they were able to sort of show a DNA effect, I think the effect on the minichromosome and that multiple pathways of re-entry seems to have been not achieved at the doses so far studied. So my last targeting strategy will be in surface antigen. Hepatitis B surface antigen clearance is an endpoint of therapy, and as we've discussed, decline in these surface antigen levels has been proposed by a number of individuals as an important immunological reconstitution step. Is it involved in exhaustion? And so several strategies have been in evaluation. We've just discussed the RNAi story. We will hear from the nucleic acid polymers, and I think antibodies also may have a role as well. I think it's just worth reminding ourselves the enormous burden that the chronically infected host has to wear in terms of surface antigen. Now, this was exploited positively in the 70s in terms of making the first generation hepatitis B vaccine. But just to remind you, I still get staggered even at this stage the amount of protein that is made in a chronically infected individual, over almost 0.5 milligram per mil typically, and this representing 1% of total serum protein. So the oncotic effects are amazing. But having said that, the other thing is that the little bit of protein chemistry I understand is that it's populated with a very large number of cysteines. Here's the cysteine raft shown for you here, and similar number of purulines, which put in perpendicular kinks into the chain. So this is just the weirdest structure, and it's got 4, probably even more transmembrane domains. So in an infected individual, if you look at the electron micrograph here I've shown for you, the subviral particles, these 22 nanometer particles, are very pleomorphic. They're not homogeneous also. And so this virus has developed a number of strategies where not just the virus is weird, but also the subviral particles are pretty strange as well. And then it has these filamentous forms. So as we discussed in the debate last night, the virus sort of makes sure that even on integration, that the surface antigen is still produced in huge excess. And it's very hard to discriminate. It's actually impossible to discriminate episomal versus integrated surface antigen. And this is one of the virological challenges that we face. But there's a number of studies have shown that this burden of surface antigen really puts a load on TOL2, TOL9, and interferon alpha signaling, as well as adaptive responses as well. And we're seeing more and more studies showing tantalizing but not conclusive evidence of reconstitution immunologically as the surface antigen levels decline. So putting all this together, now I think Marion Peters captured the essence of the moment when she said the immunologist can leave the room. At least I've invited you back with a question mark. But certainly this virologist believes that a potent nucleoside analog is important to prevent spread and also cccDNA re-amplification. I think we will need to have some sort of a selective and safe agent to reduce or silence cccDNA. Whether we put it into the bank or whether we actually try and purge it, that's really, only time will tell. But really, the virus has learned how to sort of, through its protein, through its X, core, and S, very, very cleverly manipulate the host that it's come accustomed to. So anything that really reduces the protein burden will, I think, be a positive benefit for hepatitis B. And of course, we do wait and see, with great expectation and anticipation, to see if the immunologists can beat us to the gate. Thank you very much.